TDP-43 proteinopathy is a neurodegenerative pathology present in up to 50% of aged persons with dementia. In previous work, we reported 4 distinct clusters for people with TDP-43 pathology based on age at death and neuropathologic data in the National Alzheimer's Coordinating Center neuropathology data. Cluster 1 was enriched for FTLD TDP and FTD clinically. Cluster 2 consists of older people who were late cases without severe neurotic amyloid plaques. People in clusters 3 and 4 had high levels of AD neuropathologies. The differences between these two clusters were that the people in cluster 4 died at a younger age and had more Lewy body pathology. In this study, we investigated the differences in genetic features among the TDP-43 clusters based on biological pathway-specific polygenic risk scores. Data came from the NAC neuropathology dataset linked to the Alzheimer's disease genetic consortium genotype data. The genotype data were imputed with TopMed imputation server. Of 495 people with TDP-43, 258 people had the ADGC genotype data. We obtained gene ontology terms listed for Homo sapiens and mapped to gene to each of the GO terms. Of 18,826 GO terms, we extracted 10,728 that were constructed of three or more genes, including 7,300 biological processes, 2,206 molecular functions, and 1,222 cellular components. We also obtained AD-related GWAS summary statistics from concourse studies. We then extracted synapses whose p-values were less than 0.5 from the list. We examined the association between biological pathway-specific polygenic risk scores and the TDP-43 clusters using Kruskal-Wallis non-parametric test. In biological process, the top pathways were negative regulation of lipoprotein lipase activity and cysteine transmembrane transport. However, these pathway-specific polygenic risk scores were not significant differences among the clusters after Bonferroni correction. In cellular component, polygenic risk scores of action and cytoskeleton of presynaptic active DOM were significantly different among the clusters. In molecular function, these pathway-specific polygenic risk scores produce the largest differences. However, the differences were not significant after Bonferroni correction was applied. Disruption of action and cytoskeleton of presynaptic active zone were significantly associated with the TDP-43 clusters. Because cluster 2 and cluster 3 had a different polygenic risk score distribution, AD and late may have different genetic profiles. Investigating genetic architecture of late and AD is a crucial step toward improving clinical trial and generating successful treatment strategies.